This lecture will be about next-gen sequencing. So before you try to listen to this lecture, please make sure you have reviewed all the information on basic Sanger dideoxy sequencing, because this information expands upon those very basic ideas. One of the ideas is that there are dideoxy nucleotides. So these are particular dideoxy nucleotides that are used, and or even deoxynucleotides that are used. And one of the important things they have is they have fluorophores attached to them. And these fluorophores are cleavable by particular enzymes, but upon excitation with a certain wavelength of light, they will fluoresce. And so this is going to become important later in the presentation. So, as a basic overall idea, what happens in next-gen sequencing is that there are DNA molecules that are anchored to a surface, and they're amplified many times, and then after they're amplified, so that each little space on this microscope slide has many, many copies of all the same DNA fragment, one after the other, then sequencing is done. A form of Sanger dideoxy sequencing is done with the different adapters and the sequence, and the 4 bases are put in, and they're all fluorescently labeled so that we can tell whether it's a G, an A, a T, or a C in there. So overall, what's happening, it used to take days and days to weeks. So it used to take maybe 10 days to get a good complete sequence out of them. But in the few years since I've started giving this lecture, there have been increases in the ability to perform in a shorter period of time, and so all the timelines have gone down. So it used to take, almost an entire day to do the preparation of the DNA, and now that can be done in an hour and a half. So that's how the timelines are getting much, much shorter. So as an overall plan, the first thing that they're doing is they're fragmenting the DNA that needs to be sequenced. Now, I will tell you that you would not use this if you just have, say, a plasmid that needs to be sequenced. This procedure's expensive enough, and sequencing short reads of DNA is inexpensive enough that you would only use this if you needed to do entire genome sequencing on somebody. So you're going to fragment the genomic DNA that you're interested in sequencing, and when you fragment it, you might leave some sticky ends. So you're going to repair the ends and then add an A overhang onto it so that you can ligate very specific adapter sequences, which are going to allow the DNA to bind to those little slides. And then you're going to select a size range of the ligated DNA. And the next thing you're going to do is take that size range of DNA and you're going to hybridize that to these little flow cells. And I have a picture coming up. And then the amplification is going to be done with that. And then they sequence one strand and then they sequence the other strand. So this is the big picture of what's happening. So it can be done now in as little as basically 1 day. But for more complicated things, it still might take up to 6 days, depending on how many iterations, sorry, how many iterations you need to make sure that you have the entire genome sequenced for someone. So this is what the flow cell is, and it's like a large microscope slide. Larger than what you'd think of as a microscope slide, but still, probably less than 4 inches long. And the surface of this microscope slide has 8 different channels in it. So this will enable you to sequence either 8 different people's DNA if you need shorter reads of the DNA, or if you needed to do whole genome, you could load someone's DNA in all 8 channels so that you get maximum overlap of all the sequences that you are trying to sequence from the DNA. And this surface of this microscope slide is coated with these specific oligonucleotides. So in alternating rows, each different sequences of these different oligonucleotide pairs, each with very specific sequences. So again, what I said was going to happen is the DNA was fragmented and it was then blunted, so you have blunt ends, and phosphorylated so that you can add a specific A onto the end. And adding this A gives a place for this adapter, which has a T right here, to sort of hang on to while the ligase is working on here to join these DNA sequences together. So then you have a very important adapter right here as part of your DNA sequence. So you know the sequence of the adapter, you don't know the sequence of the DNA that's in here. And then PCR is done so that you have just a short number of cycles so that you get potentially more than one copy of each of the fragments that you're trying to sequence, again, so that you can have maximum overlap, particularly if you're trying to do somebody's genome, and a certain size range is selected that's going to work best on these microscope slides. So they are then put into this piece of equipment, cluster generation station, and here's what it looks like. That flow cell is underneath the clamp right here, and the DNA is in little tubes, and it is pumped across this flow cell by the equipment. So each of these, and these can be from the same person or different people, each of these has some aqueous solutions of DNA going across that microscope slide. And basically what's happening next is you're taking the single molecules which were started off attached to the slide, and you're going to make many copies of the single copy of DNA that's going to attach onto that. So here's how that works. So in parts of the microscope slide, remember that you have your unknown sequence of DNA right here, and you have adapter sequences on the end of the DNA. So you want each of these to be very well separated from the others, so your DNA cannot be too concentrated as you move it across the slide. So it then binds this DNA with the adapters. The adapter causes it to bind to the sequence that's on the microscope slide, but well isolated. And then the next thing that happens is a polymerase comes up and extends this sequence all the way up. So now you have a double strand of DNA. This strand right here is actually not covalently connected to anything. It's only being held together with hydrogen bonds, but because this little sequence served as a primer for the polymerase, now this strand is covalently attached to the microscope slide. So they then discard the original template, and your newly synthesized strand, which is covalently attached now to the microscope slide, is going to form the basis of what happens. So again, remember that you want everything to be well separated, so you don't want one thing to be here and another thing to be right here, for example. So you want one sequence that starts in the middle of this tiny little dot on the microscope slide, and you're making multiple copies of the DNA that's going to attach to all these to fill in a small space right around here on the microscope slide, and I call it a microscope slide, but it's actually a flow cell. So you want the flow cell to fill up with all the same basic sequence of DNA, all in this very small area. So the first thing that happens is, remember you had the other adapter on the other end. You had your unknown sequence right here, and you had the adapter here, and you had the adapter right here. So the other end of the adapter is going to be able to bind to one of the other sequences, the other oligonucleotides that are attached covalently to the microscope slide. So this DNA sequence bends over, and that's, remember, we selected for a particular size range of DNA, and that's so that you can have this bending happen in the right orientation and the right order. So it bends over to the sequence next to it and binds to it, and then this forms the next primer right there for polymerase to come back around and extend and make this double-stranded. So now you have double-stranded DNA here, and then you're denaturing it. So now you have one strand here and one strand here. They're opposite sequences from each other because they were originally anti-parallel to each other. So you have two copies of the DNA, one from each original sequence of the DNA, 5' to 3', and the reverse. Then the process happens all over again. So this one bends over this way, this one bends over this way, and again makes them both double-stranded, and it keeps going and going and going till you have multiple copies, all in that tiny, tiny area on the flow cell. And so you've denatured all of them. So half of these are one sequence, and half of these are another sequence. So you start by saying, okay, we're going to cleave all of the reverse strands. So after you cleave all of the reverse strands, now you have only forward strands on the DNA. And then, remembering that you have an adapter on this end of the DNA, you're adding a sequencing primer onto this little strand of DNA. And that sequencing primer is going to allow a polymerase to extend the DNA. So all of these are exactly the same sequence right here. So even though you started with basically only a single copy of one of the strands of DNA, now in that very small space on the flow cell, you have multiple copies of that. So here's what's happening with that sequencing primer. So here's the original DNA strand and the sequencing primer that's binding to part of that adapter that was added on. And one at a time, it says, okay, so the next base here is an A. So I'm going to need to add a T right here. So it adds a T right here. And it just adds a single base. So unlike in PCR, the polymerase is not adding multiple bases. So it's adding a single base right here, and then it's hit with a wavelength of light, and it fluoresces, and it fluoresces a particular color because you have a particular fluorophore attached to each of the G's, A's, T's, and C's, a different fluorophore for each one. So remember that I told you you should know the difference between processive and distributive enzymes. So this polymerase that we're using here, this is a distributive enzyme where one single base is added, and then the polymerase stops after only adding one base and detaches. And this allows the rest of the procedure to occur. It allows the fluorophore to fluoresce, and then that fluorophore is chemically cut from the base, which then makes a space for the next base in line to be added by the enzyme, and the next one in order is a C right here, so a G is added right here. And again, that G is now going to fluoresce green. So each of them in turn are added in there. So you can, doing this, you can add about 36 to 75 bases. So very short reads of the DNA. When you're doing regular Sanger dideoxy sequencing, as I show in the other videos that I posted, you can get reads of up to 600 or 800 bases from one sequencing. But in this one, because you have massively parallel systems, you are simply doing very short reads and using computer memory to overlap all of these short sequences so that you can get one complete sequence of the entire genome that you're looking for. So on each strand, you're only interested in doing this maybe around 75 times to get very short reads of the DNA. So here's what it looks like. So this is kind of what the flow cell looks like, and what's going on over the flow cell is there's a very high resolution microscope lens over it, and that microscope lens is really able to zero in on each of those individual clusters, and it just sort of goes down the slide, and the computing power remembers where each of these clusters are, and it will say, okay, last time this cluster was red, and this time it's green, so it'll say it's a T, and then it's a G, and then it'll be yellow, so it'll be this particular base. So it remembers in its memory, the location and the color sequence that it is, so that it can assign a particular sequence to it. Now, it's not nearly as useful if you have very much overlapping sequences right here. So, or something like this. This is pretty useless. So it's only going to remember and give you the sequence for the ones that are well isolated on the microscope slide. Some of them are going to be closer, and some of them are going to be farther away from everything, and those are the real useful ones. But because you're amplifying the single DNA that binds in the middle here, you're getting around 1,000 copies of the DNA in every cluster, and that's enough because all of these are going to fluoresce the same color at the exact same time. That's enough for the very high resolution microscope and very high resolution camera to pick up. So overall, on all 8 lanes of that flow cell, you can have around 150 million different clusters. So 150 million different short sequences of DNA. So you can basically have someone's entire genome represented within those 8 lanes of that flow cell. Now, other companies have come up with similar, different ways. Illumina. So this is the methodology that's used by Illumina. Illumina is far and away the technology leader in terms of sequencing. They're head and shoulders ahead of the rest of the crowd in terms of the number of instruments that they sell and the popularity of their system, which is why I've chosen to emphasize the Illumina system in terms of explaining next-gen sequencing. Other companies have come up with, similar, but because of patent rules, slightly different ways of doing it. So, for example, Lycor Biosciences, they are using fixed enzymes, and then they put the DNA through the enzymes that are fixed in position. Or Roche starts off with the same idea of a microscope slide, except instead of a slide, they have little beads. And so you have one DNA piece attached to each bead, and then that's amplifying along the surface of the bead, and then you're able to break the connection to the bead and break and have the bead be fixed to the microscope slide, all with the exact same sequence on it, and so then it's all going to fluoresce because it's all on this one bead. So it's closer to this idea than Lycor. Lycor is really third place because this doesn't work nearly as well as this or even the Roche method. So whatever method you're using to do all these individual little short reads of the DNA, the computing power, and there's a lot of computing power in there, is able to say, okay, well, this piece overlaps this piece, which overlaps this piece, which overlaps this piece. And so basically it can say, okay, well, here's an overlap, here's an overlap, here's an overlap, and it's going to align all these short, maybe, 50 to 75 base fragments and put them all together in its computer memory so that you get one long aligned consensus sequence, and that is what can be reported as the sequence of what you are trying to sequence. So here's an example of the different types of instrumentation that Illumina sells. So if you have smaller needs, like, for example, 23andMe, they want high throughput. They want to be able to do lots of samples, and they want to be able to do them very quickly. So it has a very short run time. And when you're doing with a company like 23andMe, they're not actually sequencing your entire genome. They're mostly interested in some short specific segments of your genome so that they can match them up with their proprietary information of this particular short stretch of the genome, or this, this particular base change is associated with this type of genetic heredity. So short reads, are fine, and having a fairly large number, but basically they want a very short runtime. On the other hand, if you are sequencing someone's entire human genome, population scale sequencing, you're willing for it to take a longer number of days and to have more reads available in a flow cell. So you're going to have much more information that's generated for a single event, but you're going to use a different type of instrumentation. So Illumina has all these different types of sequencers, some of which are used in doctors' offices for doing genetic sequencing if they suspect genetic anomalies, and some of them are used just to say, do you have a breast cancer marker type gene? So they're used, the different outputs, the different speeds are used for different kinds of tests. And that's it.